The nucleus is a large, dark sphere housing our genes, which house our hereditary characteristics. Most cells have one. Red blood cells don't have one. Some have multiples like muscles because as they grow in length, they are fused smaller muscle cells together, so the nucleus stays behind, so they have multiple ones when they're grown up. A cell must have at least one of them to be able to divide. Two elementary membranes filled with pores create a boundary to the cytoplasm. The nucleolus on the inside of the nucleus is a factory for ribosomal RNA, which basically are ribosomes. We'll talk about those in a little bit. And this brings us to the chromosomes and our genes. Chromosomes are the carriers of our hereditary characteristics. One gene contains information to make one protein. The chromosomes, therefore, are the cookbook for proteins. We have 46 chromosomes, two sets of 23. 23 maternals from the mom and 23 paternals from the dad. This makes us a diploid. Haploids, in contrast, have only one set, not two sets of chromosomes. We humans have 30 to 40,000 genes packed in a DNA double helix of about 2 meters in length. That's about 2 yards. That's a lot. This needs to be very well organized, otherwise we have a mess. So this long strand of double helix will coil around histone proteins. They wrap particularly tightly when we are doing mitosis. So what you see then are these large X's, and where they cross in the middle is called a centromere. If we need to make protein, we uncoil the part of the double helix cookbooks that holds that specific gene. Let's talk more about how the genetic code works. This is the setup. DNA is 2 nucleotide strands connected by hydrogen bonds into a double helix. 3 nucleotides in a row code for 1 amino acid. We call that a triplet or a codon. Chains of amino acids then make our proteins. How do we get them made from DNA? Well, we'll have to first find the right gene. Then we unwind and unzip the double helix at that point. Now, since the DNA will not leave the nucleus because that would be too dangerous, we make a copy of the nucleotide strands that codes for the specific protein. This strand now is then called an mRNA. It leaves the nucleus, feeds into the ribosomes, where the nucleotides are decoded and proper amino acids are brought and attached to one another. Well, let me explain RNA a bit more. It is similar to DNA, except it has only one strand, it has a different sugar, and it uses the nucleotide uracil instead of thymine. I know it's complicated. That's why we're doing it this way. We have 3 types of related structures that we call RNA. Messenger RNA or mRNA is the copy of DNA. Ribosomal RNA or rRNA is the part of the ribosomes that is reading the nucleotide codons. And transfer RNA or tRNA brings the proper free-floating amino acid from the cytoplasm and helps attach it to the growing polypeptide chain. Let's do it again because we have 2 more new terms that we need to feed in here. So first, we uncoil and unzip the double helix at the appropriate gene. Then we copy that using RNA polymerase, an enzyme. This process results in the mRNA strand and is called transcription. That's a new term. Transcription. The mRNA then leaves the nucleus through a pore and feeds through a ribosome where tRNA brings the proper amino acids and attaches them in the proper sequence. This process is called translation. I know, it's a lot of terms. When transcribing, we transfer the information to a different medium from DNA to mRNA. In translation, we change the language, so to speak, from a genetic material to a protein. That's how I remember it. Maybe it helps.